occipital lobe. This lobe is present in the posteriormost aspect, okay, and this is essential for reception and recognition of visual stimulus. Any visual stimuli that's coming from the eye, it will receive it and recognize. So, for this purpose, the reception and recognition of the visual stimulus, this occipital lobe is divided into primarily into the visual cortex and visual association cortex. The primary visual cortex and visual association cortex. Second is visual association cortex. So, primary visual cortex is also known as striate cortex or striate area, and this is area number 17. And this visual association cortex, they are also called as, instead of called striate, they are called as extra striate, and they are area number 18 and 19. So, primary visual area is area number 17, and secondary visual area, or not the secondary, the visual association area, they are area number 18 and 19, okay, area number 18 and 19. Now, this visual cortex, that is the primary visual cortex, the area number 17, where exactly it is located? It is located on the medial aspect, medial portion of the occipital lobe. On, say, if I try to draw the medial aspect of a particular, like say cerebral hemisphere, then there was this sulcus which was dividing the occipital cortex into the cuneus and lingual gyrus, right, cuneus and lingual gyrus. So, that sulcus is calcarine sulcus. So, this area number 17, primary visual cortex, it lies on the medial portion of the occipital lobe on either side of the calcarine sulcus, okay, and its major afferent of this pathway will be coming from, see, they are coming from the both the eyes, optic radiations, but the gateway to any sensation is thalamus. So, which part of the thalamus is concerned with visual sensation? The lateral geniculate body. So, from the lateral geniculate body of the thalamus there is input, and these input fibers which are coming from the lateral geniculate nucleus, some input fibers, they are gathered into a thick bundle, they form a thick bundle, okay. This thick bundle, that can be visible if we do a cut section, gross cut section of the brain, we can visualize that thick bundle, and there is a name for that thick bundle, that is called as line of Gennari. In ANAT, the ANAT people will teach you about this thing, GENNARI, line of Gennari, okay. And see, like the representation of point to point representation of the entire retina is present, retinal surface is present in this area number 17. So, that if there is a damage to a discrete, say this is very large area, like around the calcarine sulcus, almost all that part. So, if there is, suppose there is a lesion happens to only this much part, this part, or say this dot, this particular dot, or this particular dot, or this particular dot, then there will be loss of the vision in that particular representation of the fiber that's coming from a particular part of the retina, right. Say this part is represented in the retina over here, this part is represented in the retina over here. So, this is the retina, and in the field of the vision, this part will have the image from this area, and this part will have image from this area. So, if there happens to be any injury at this particular area of the area number 17, this lesion, then that person will have the disability in the field of the vision at this particular point. So, this point is called as scotoma. What is this called as? Scotoma, or scotoma means a particular blind spot. So, there will be a blind spot in the corresponding portion of the field of the vision which is getting represented in the lesioned part of the primary visual cortex. So, that a smallest area, that scotoma, will be there in the corresponding portion of the visual field. A unilateral lesion in area number 17, say one side, either the right or the left, is getting affected, then what will be the pathology? That pathology will be called as contralateral homonymous hemianopia. These you have read in your 11th and 12th standard, and you will read in the vision also. So, I will not go into detail because the special chances you will read about these things. Contralateral homonymous hemianopia. If there is total absence, then it is called as hemianopia, otherwise hemianopsia. Okay, hemianopsia. So, with macular sparing, there will be one term you will be reading about it, with macular sparing. Why the macular sparing? Because the representation of the macula in the cortex, in the occipital cortex, is at such a particular point which has got dual blood supply, middle cerebral artery and the posterior cerebral artery. And most of the times, as I have mentioned previously, I guess, like the lesions are due to the either the blood supply is getting hampered due to either the stroke, any thrombus formation. So, there is compromise in the blood supply. So, if that compromise happens to be only in the posterior cerebral artery, then the entire of the visual cortex will get affected, and only the area which is representing the macula will be spared, because it has got the dual blood supply. There is compromise in the blood supply coming from the posterior cerebral artery, but still the middle cerebral artery blood supply is there. That is why there will be the macular sparing in that contralateral homonymous hemianopia. Okay, contralateral homonymous hemianopia. Now, if there is bilateral lesion instead of being unilateral, if the lesion happens to be the bilateral, then that pathology or that thing deficiency is called as cortical blindness. That person is suffering from the blindness because the bilateral lesion is there, cortical blindness. And in this cortical blindness, the patient cannot see, patient won't be able to see, but all the other reflexes related to the eyeball, like the pupillary reflexes, okay, those sphincter pupillae and all these things, that will be working properly. The oculomotor nerve, all those nerves, they will act properly. So, the person will not be aware about that he is blind, because all the sensory modality are intact. So, what he will do? He will deny the blindness, and he will try to say, you will say, like you have got the issue with the your vision bilateral, like cortical blindness is there. So, you will ask what is there, like what is in your field of vision, you will put something. Then because he has got the cortical blindness, he is not able to tell the object, he will try to confabulate it, means वो झूठ बोल के कुछ ना कुछ बोल देगा, कुछ भी ऐसे ही. That is called as confabulation. So, deny the problem and will try to confabulate it. Okay, confabulate it. So, that is regarding the bilateral lesion within the primary visual cortex. Now, the visual association cortex, these visual association cortex, these are these areas, the area number 18 and 19, they are distributed throughout the entire occipital lobe and a bit of throughout the occipital lobe and a bit of the posterior part of the parietal as well as temporal lobe. So, it is a diffuse distribution of area number 18 and 19. Okay. So, these regions, these area number 18 and 19, they receive fibers from primary visual cortex, that is the striate cortex, the area number 17, and they integrate these visual input. They integrate the visual input from both the hemisphere, from both the hemisphere, and from the retina to the visual, these visual association areas, information is like, say, you will read about when you will go through the retina. Let me tell you these few things about this one. See, any object when it is visualized, what we visualize? The shape of the object, right? The shape of the object, the color of the object, shape of the object, color, form of the object, right? Form of the object. Then if there is any movement or motion, like, right, and depth of any particular field. Okay. So, all these like shape, color, form, motion, depth, and the spatial, like what is the location in the space, this spatial information about that particular object. So, all these like characteristic feature of an object, they are perceived by two different system. One system is called as cone, other one is called as rod. So, the cone stream, this cone stream, it originates mainly in the, say this is the retina, it originates mainly from the central part of the retina. And this cone stream, see, there will be, you will read about these cells in the retina. There are two type of the cell, the magnocellular, larger cells, and parvo cells, magnocellular and parvocellular pathway. So, this magnocellular is larger, parvo is smaller. So, this cone stream, it originates from the central part of the retina, and this is formed by the parvocellular pathway. So, and this cone stream, this is concerned about the form of an object, color of an object, cone stream, and that's getting originated from the central part of the retina, It relays through different, like say there is point by point representation of the retina within the cortex. So, that point by point representation of the retina is not only present in the visual cortex, there is point by point representation in the lateral geniculate body too. So, these tracks, the fibers which are forming the cone stream, they will relay through a separate layer from the lateral geniculate body, and they will project to the primary visual cortex, and from the primary visual cortex it will come to those areas, visual association areas 18 and 19, as well as see, there is one more term that is called as blob cells. So, these cells are a particular type of the cells, they are present in the area number 20 and area number 21, and in the primary, this one visual association area 18 and 19. So, this parvo cellular pathway, this is called as parvo stream, is ending up on those blob cells. So, that is the parvo blob system, parvocellular blob system, that's ending on the visual association area number 20, the 18, 19, as well as area number 20 and 21 in the inferior part of the temporal lobe. So, if there happens to be any lesion in these visual association areas or area 20 and 21, then it will lead to one term that's called as metopia or metopsia. Metopia is if there is no S, then there is total absence. Opsia means there is loss but not the complete absence. Chromatopsia, achromatopsia. So, color, if there is total absence, then it is called as achromatopia, complete loss of the color vision. If there is lesion in the right sided, these association areas, area 18, 19, 20, 21, then if there is a lesion on the right sided of these areas, then there will be the complete loss of the color vision from the contralateral hemifield. left side aspect of the field, contralateral hemifield. Patient will be able to see everything, there won't be any issue, but they will be in the shade of the gray, there won't be any color. Okay. And because I said the fusiform gyrus, this is also the nearby area, that is why if the person is having these lesions, will also suffer from the prosopagnosia, inability to recognize the faces. Now coming to the second part of that characteristic feature of a particular object, that was the motion and depth. This parvocellular is concerned about mainly, see, there are so many things. I said there is color, there is form, there is motion, depth and spatial. So form and color is for parvocellular. Motion and depth regarding any depth of that object, like 3D, we can say the 3D or 4D, that is concerned concerned with the magnocellular pathway. Magnocellular is for M for magno, M for motion and depth, magnocellular, and parvo was for form and color. Parvo was for form and color, parvo. Okay, this one. So the motion and the depth, they are processed by the magnocellular. Like say this one parvocellular, I will depict it with red part. That was that parvocellular pathway, parvocellular cone stream was originating from the central part. Now this remaining part, the peripheral part of the retina, instead of the cone stream, it will be rod stream, rod cells, rod stream. Okay, rod stream. So it originates in this peripheral part of the retina, in this peripheral part of the retina, and again it will, there will be the point by point representation of this peripheral part of the retina of the magnocellular pathway or the rod stream in the lateral geniculate body, right, lateral geniculate body, and it will project to the primary visual cortex in the these areas where this part like ends up in the primary visual cortex. They are called as thick, they are the thick striped zones, striped area. Okay, striped zones, thick striped zones, and these zones, these areas, they project from the primary visual cortex to the temporal lobe and parietal lobe, area along with area number 18 and 19. See, I said there is no, like, it is not like occipital lobe will work separately, it is working in according, mainly there is like the function of this visual sensation, perception of the visual sensation is occipital lobe, but it is also working in cohort with temporal as well as the parietal lobe. Okay. So if there happens to be the lesion in this stripe zone or associated from the stripe zone, there is, see, that cone stream was also relaying in area number 18, 19, 20 and 21, and this one, the rod stream, is also relaying in area number 18 and 19. So those relay area for these two different stream, whether the right stream or the like the rod stream or the cone stream, they are separate parts. So if the part of the area number 18 and 19 which is receiving the rod stream or the magnocellular system pathway, if it gets affected, then that person will suffer from the deficiency in depth and motion, like there won't be three-dimensional view for that person, though the color and form will be normal, but depth perception will be lost. Okay, depth perception will be lost. So that's like if there are any this is the clinical significance of the visual cortex and the visual association cortex, and we have covered this visual agnosia, like, right. So visual agnosia is if the involving part of the occipital lobe as well as temporal lobe both are getting affected, that person won't be able to recognize visual pattern, though the visual field defect won't be there. Like say, what do you mean by visual agnosia? Visual agnosia, like what is the difference between the visual agnosia and blindness? Blindness is not able to see the object from the field, and visual agnosia is the person is able to see the object perfectly. Let me give you an example. Say if you show a person a spectacle, okay, like if you show the spectacle, that person won't be able to name it that this is spectacle. He is able to see it. So if you ask what is it, he won't say it is a spectacle, he will say it is a thing which is made up of two circles and there is a bar that is connecting these two circles and then there are two more bars. I guess this example clarifies what is the difference between the visual agnosia and the blindness. In the blind person won't be able to say like there are two circles or these things, he won't be able to see anything. But visual agnosia is visually he is not identifying that visually. Like say अगर आपको कोई स्पेक्स है, स्पेक्स आपको हाथ में दे दिया, आपने आंख बंद कर रखी है, हाथ में पकड़ा दिया गया, आप उसको फील करके बता दोगे, दैट इज कॉल्ड एज स्टीरियोग्नोसिस. इफ यू आर नॉट एबल टू टेल इट देन इट इज कॉल्ड एज ए स्टीरियोग्नोसिस. ओके राइट. एंड अब उस चीज़ को आपने जस्ट सामने रख दिया, विजुअली परसीव करवाया गया है, तो विजुअली ही बता रहा है तो वो विजुअली इंटैक्ट है, विजुअल सेंसेशन से देख के बता दिया दिस इज ही हैज़ लर्न दिस एक्टिविटी, this is specs. Okay, but if there is issue with this particular part of the occipital and temporal lobe, visual agnosia, then that person won't be able to name that object like this is specs. He will say ये circle का बना है but won't be able to say it ये चश्मा है. Okay, so by this we have covered all those lobes, all these different lobes. So what in conclusion I can say, like say the parietal lobe जो हमने कल किया था, parietal lobe is like if the person who is having any issue with the dominant parietal lobe, he was suffering from the just man syndrome, and the just man syndrome was and the person will have symptoms, won't be able to write the things, won't be able to understand the written things, alexia, agraphia, then there will be finger agnosia, finger anomia, not able to name the fingers, not able to differentiate between the right and left, not able to do the calculation. So this proves like how much important when these things are inculcated in our life. जब हम स्कूल में जाते हैं, केजी में होते हैं, तो हम जो सीखते हैं, काउंटिंग सीखते हैं, हम सम करते हैं, सब्ट्रैक्शन करते हैं, ये सारी चीज़ें सीखते हैं, तो हम स्टार्टिंग में फिंगर से करते हैं ना, उंगलियों पे गिन के कि 1 2 3 4. सो दैट इज़ द मेन पॉइंट, दैट इज़ लाइक से व्हाट इज़ द प्रैक्टिकल इम्प्लीमेंटेशन ऑफ ऑल दीज थिंग्स वी आर टॉकिंग अबाउट नाउ. दैट इज़ द प्रैक्टिकलिटी, like we use the finger at the very early stage of our life to name, to understand the things, to calculation, all those things by using the fingers. Okay,